Dr. Sota Koeda, Associate Professor, Kindai University, Nara, Japan. He will be delivering a lecture on Begomovirus resistance in hot peppers and its application to breeding. He is presently working in Kindai University, Japan, and his area of work is breeding vegetables for resistance to Gemini virus, Begomovirus, and poliovirus. I request Sota Koeda to please address the gathering.
So, first of all, thank you very much for inviting me to this interesting congress. And today I would like to introduce our recent research related to Begomovirus resistance in peppers. And final goal will be breeding new cultivar resistant against Begomovirus. Okay, so maybe all of you may know, but in around the world, more or less 42 million tons of peppers, capsicums are produced, and five big producers are like China, Mexico, Turkey, Indonesia, and also India is a big producer. And most of these countries suffer the Begomovirus core disease. And we started this research around 2010, which is more or less 15 years ago. And this photo was taken when I first visited Indonesia. This is kind of typical pepper growing field. And if we get close to plants, we can see the viral disease like this in almost all plants. So initially, I didn't even know Gemini virus or Begomovirus, but I could understand that the fruits cannot be harvested from these plants. And because there are no pesticide for viruses, farmers cannot earn money if their pepper plants are showing disease like this. So after coming back to Japan, I searched for papers and found out that this Gemini virus called disease in peppers is not just a problem in Indonesia, other countries also. And 15 years ago, there was not so much progress in this research area, and also there was no commercial varieties. So I chose this as a research topic. So now Gemini virus family contains 14 genus. One of them are Begomovirus, which is the largest one, and Begomovirus contains more or less 450 virus species. And Begomovirus is transmitted by whitefly. So the basic disease control method is to treat insecticide. And also when we cultivate plants in greenhouse, farmers could find mesh around the greenhouse to prevent the contact of whitefly to crops. But because most of the pepper plants are grown in open field, this basic method is not so much efficient to control the disease. So this is why people need virus-resistant varieties. So there are several hotspots for Begomovirus core disease. One is Southeast Asia, one is South Asia, which also include India, Pakistan, Bangladesh, and also West Africa, Central America, and South America. So there are different Begomovirus species, but basically they induce very similar symptoms, like yellowing or coloring symptom. And in case of India, you have so many Begomovirus species, so I guess it is almost impossible to completely understand the situation. But anyway, we can say this is a global problem in pepper production. So initially, we started our work in the field for screening resistant material. But if we did this type of work, we faced difficulty because in the field, not only Begomovirus, but other virus like other RNA virus or other pathogen, or plants get stressed with heat or water. So everything is mixed up. So we decided to choose different approach. So for this, first we did research in climate control condition in laboratory condition using small seedling and also pure virus isolate, single virus isolate, and using the infectious clone for efficient inoculation. And these are all for accurate phenotyping of Begomovirus resistance because now we have NGS technique. Genotyping is not a big problem. I think phenotyping is the most important one. So for our research, we divided into four different stages. In stage one, we visited Indonesia several times and did field survey to identify the virus. And then we did the inoculation method and did the screening. And in the first stage, we identified several resistant genes in peppers. So to make long story short, I think the design for the infectious clone is significantly important because most of the Begomovirus species which we use for our studies are bipartite Begomovirus, which has DNA and B. So initially we cloned the full length and partial length DNA and DNA-B in separate plasmid. But this will work nicely in Nicotiana, Benzamiana, or tomato, but not in peppers. So we clone both DNA and B in the same plasmid. And by doing this, we successfully inoculated Begomovirus to pepper. This is artificially inoculated one, and this is the naturally inoculated one in field. So using this technique, we screened like more or less 900 pepper accession, which contains all pepper cultivated species. So we screened two promising material. One is DAPF5, which is capsicum onion, which we found in Indonesia, local accession. And another one is PG11, which we introgress resistance from capsicum pubescens to capsicum onion. So we have already published these two genes. And let's start with BFF5. So when we inoculate Begomovirus to BFF5, the plant is almost symptomless, but the susceptible pellet BFF4 is showing typical yellowing and also F1. So we could say this is a recessive resistance. And when we check the viral DNA accumulation of Begomovirus in this plant, in susceptible BFF4 and F1, there is a lot of virus accumulating, but in BFF5, its accumulation is significantly restricted. So we would say BFF5 has a resistance against at least two Begomovirus species. And we prepared F2 population. Here we inoculated around 500 plants and we selected the virus positive plants, more or less 350. And we phenotyped susceptible plant and resistant plant. And we also checked the viral DNA accumulation in each individual. And using these as a phenotype data, we combined with sequence data to map the resistant locus. And we detected single peak on chromosome 5. And because it is a recessive gene, we named this small pepper 1. And in this case, we just added DNA marker, and we are very lucky that we finally narrowed down the candidate lesion to 34 kb, which contains just one gene. So this gene encoded a protein called pelota, and there was a single SNP A to G in resistant line BFF5. So this SNP located in the ninth intron of its splicing region. So by this SNP, ninth intron was not spliced out, but actually it was translated to amino acid. So in the pelota protein of BFF5, there is 28 amino acid additionally. And from our recent study, we found that the function as a pelota is significantly decreasing in this resistant line. And in the end, we conclude by virus-induced silencing. In this case, we use a susceptible genotype. And when we check the control, when we co-inoculate TRV and peptide CIV Begomovirus, it's still susceptible. But when we silence pelota using BIGs and inoculate Begomovirus, it became resistant. So we concluded that small peptide-1 gene is encoding pelota. And also we developed a DNA marker on SNP, in this case CAPS marker, so we can distinguish the resistant genotype and also susceptible genotype, also heterozygous genotype, which is susceptible. So these markers are very useful. And we did similar thing also in PG11. So I should make the story short. So here PG11 is almost symptomless, although infected with Begomovirus, and this one is a susceptible control. And in this case, the resistance is dominant trait. It is different from the previous one. And when we check the viral DNA accumulation, it's nicely restricting the accumulation. So we would say it is also resistant to Begomovirus. And we mapped the gene on chromosome 7, which is different. And we found out it encodes RNA-dependent RNA polymerase, which is RDR. And we developed an engine marker. Here we can distinguish the resistance to susceptible one. And in the end, we did bigs again. In this case, we use PG11. So in the control plan, it's still resistant. But when we silence RDR expression by pig and inoculate Begomovirus, it becomes susceptible. So we can say that peptide-2 gene is encoding RDR. So in case of peptide-1 material, we finish the field test also. In this case, we cultivate more or less 420 F2 individuals with their parents. So in case of susceptible line, BFF4 is showing heavy symptom. But as we expected, BFF5 show only mild symptom, but we couldn't control perfectly the disease. But what is important is that disease onset and progression was much slow, and at least we can harvest twice or double number of fruits. So most recently, we published this paper. It's very simple. We combine two genes, peptide-1 and peptide-2, to single pepper plant to make the peppers more strong against Begomoviruses. So for this study, we use 4 pepper genotype. One is susceptible number 218, another one is Begomovirus tolerant capsicum chinense accession GR1, and the previous BFF5 and PG11. So we check the presence of peptide-1 locus and peptide-2 locus, but with the previous DNA markers. And in this study, we use 4 Begomovirus species grouped in different 2. One group is from Southeast Asia, peptide CIV and peptide CAV. Another group is from Central America, PHYBV and PepGMB, which are also very famous Begomovirus for pepper studies. So in this case, we first singly inoculated 4 different Begomovirus to 4 different pepper genotypes. So the yellow circle means resistant, and blue X means susceptible. So in case of BA.5, it was resistant against both of the Asian Begomovirus, but not against the American Begomovirus. It was susceptible. But PG11 was resistant against all 4 Begomovirus species. And we also checked the viral DNA accumulation. So BFF5 was restricting the viral DNA accumulation of Southeast Asian Begomovirus, and PG11 was restricting the 4 different Begomovirus. although PEP-GMB accumulated little more than the other Begomovirus species. So next, we use the previous plant, which was agro-infiltrated, to make artificially the mixed infected plant, which is often seen in the field. So in this study, we inoculate two Southeast Asian Begomovirus or two American Begomovirus with four pepper genotypes. So by doing this, the result became completely different. So just BFF5 show resistance against co-infection of Begomovirus from Asia, but not against American Begomovirus, and also PG11 became susceptible. So I would say just using one gene is not enough. So BFF5 is still restricting the Begomovirus of Asian isolate only. So in the end, we checked the high-limiting effect. So in this study, we used two Begomovirus, PHYBB and PEP-GMB, which is highly pathogenic in the previous study. So first, we agro-infiltrated to susceptible number 218, and we checked the infection, co-infection by PCR, and take the scion and graft to 5 different pepper genotypes. One is susceptible number 218 and BFF5, PG11, and we produced the F2 progeny by crossing BFF5 and PG11. So F2 hetero has PEPY1 in homozygous state and PEPY2 in heterozygous state. F2 homo has both genes in homozygous state. So BFF5 and PG11 was susceptible, almost same as number 218. But when plant has PEPY1 in homozygous state and PEPY2 in heterozygous state, the symptom was much mild. And in case of the plant which has both in homozygous state, it became almost symptomless. And when we check the viral DNA accumulation, here is a homozygous one, the purple one. It is restricting PH5BB, PEP-GMB both in two different experiments very nicely. So we can control that if we introgress two different genes in peppers with homozygous state, it will have very strong resistance against Begomovirus. I think it will work well with field condition. We will see. So in case of tomato, US, Israel, Netherlands, Japanese team work hard and they have already reported 4G. In case of pepper, we are the only one who cloned resistant gene, which is RDR and Pelota. And we are also working for cucurbit and other farinaceous plants, and we have already cloned RDR from cucumber, and we are now fine mapping some gene, cucumber, melon, and eggplant. So these genes will be used for each vegetable species, but now we have modern technology like genome editing or EMS mutation, so we can apply this knowledge across different crops, I guess. So more detailed information can be found in our website or research data anyway. So please contact this site if you are more interested. So this is all from me. Thank you very much.